Hello children, welcome you all to the Form 4 Biology session. Today I am going to discuss one of your first term content, enzymes, which comes under the SOW reference 2C. Now, during this lesson, you will be able to understand what are the enzymes and how enzymes are functioning and what are the factors affecting for the enzymatic reaction, especially how temperature and pH act on the activity of enzymes. Let's see what are enzymes. All the enzymes are proteins and they are called as biological catalysts. Why we call them biological catalyst? Because they are made in living cells and also they are important to catalyze the metabolic reactions. So simply they are increasing or speeding up the rate of the chemical reaction. And also enzymes are specific to substrates. So in the enzyme structure, they have specialized sites called as active sites. So active sites are very important because the substrate we call the reactant molecules, they are coming and binding with the active site of the enzymes. So the function can be explained using lock and key hypothesis. So here the lock is the enzyme, key is the substrate. So the enzymatic reactions are affected by temperature and pH. And during the chemical reaction, these enzymes are not utilized. That means those are released. So we can reuse these enzymes for other chemical reactions. Now, as I mentioned earlier, these are the major factors which are affecting for the enzymatic reactions: pH and temperature. Now let's see how temperature is affecting for the rate of the chemical reaction, right? Normally, when temperature increases, the rate of the reaction also increases. The reason is when temperature increases, the kinetic energy of the reactant molecules, that is also increasing. So they are moving. The random motion is higher. Therefore, there is high chance for a successful collision. Because of this, the enzymatic reaction rate is higher. Up to a certain point, when temperature increases, the reaction rate and the enzymatic action also increases. After that point, the rate of the reaction and also the action of the enzymes quickly drops. Because the reason is under high temperatures, the enzymes are called as denatured. Denaturing means changing the shape of the active sites. And also under very low temperature conditions, the enzymatic rate also lesser. Reason for that is the kinetic energy of the molecules, reactant molecules, is lesser. Now you can see here, this blue color one, that is the enzyme 1, green color one, enzyme 2. Now this yellow color one is the substrate. So normally the substrate is having the complementary shape to the active site. So now this yellow color substrate is best fitted with the enzyme 1 because the active site shape and the shape of the binding site shape of the substrate, they are complementary. Therefore, this yellow color substrate is not fixing with the enzyme 2. So now you can see in the step 2, enzyme substrate complex has been formed. Then after the reactants are reacting and then the products are released. Enzyme also released at the end of the reaction. Now we will see the effect of temperature, as I told you, up to a certain point. Up to optimum temperature, the rate of the enzymatic activity increases, then quickly drops. The drop is due to the enzyme denaturation. Now this is a practical. Now we are going to check how the temperature is affecting for the enzymatic activity. So here, as raw materials, we need starch solution and iodine as a reagent, and we are using amylase as the enzyme. We can use a water bath to control the temperature. So you can see here, step 1, We are heating the starch solution up to the temperature that we want. Here we have to use different temperature values, right? Then we are taking a spotting tile which is white in color, and then we are adding iodine drops. Then after what we do? Amylase solution also we are heating up to a certain temperature like 30 degrees room temperature. Then we add amylase to the starch solution, right? So what is the reaction which happens in between starch and amylase? So the hydrolysis of starch is done by the enzyme amylase. So starch is broken down into maltose by the enzyme amylase. Normally starch shows dark blue color with iodine indicator. When starch is not there, that means it is completely hydrolyzed by the enzyme amylase. Then the iodine does not show any color. Normally the color of the iodine is yellow color, with that is the normal color of the iodine. But when iodine is reacting with the starch, iodine shows a dark blue color. That is why we call it an indicator. Indicator has the ability to change its color, right? Now here when we are increasing the temperature, what we can observe is at 40 degrees, we can observe very less amount of blue color spots. Blue color means still starch is present, but all the other spots yellow color. Yellow color means the color of the iodine. That means all the starch has been hydrolyzed. So enzyme acts fastest. The reaction rate is faster at 40 degrees and within very short time. Now we will see the effect of pH. Normally there is an optimum pH value for most of enzyme. That is normally 7. pH 7 which is neutral. But there are some enzymes which require lesser pH or higher pH for the reaction rates and to obtain the optimum yield. Right? So now we are going to check the effect of pH. In the optimum pH, the rate of enzyme activity is higher. And then when we are deviating from this optimum value, the reaction rate is getting lesser, lower, right? So what will happen at the extreme pH conditions? So at very low pH conditions, so that means far away from the optimum value. So then the enzyme is getting denatured. When the pH is further increasing, what happens? So here the active site shape is lost. Then the enzyme cannot function properly. But at the optimum pH, the active site is complementary with the shape of the substrate. So they can fix properly and then they can start the reaction. Now there is another practical to check the effect of pH. So like we have done in the earlier practical. Here also we are using the iodine solution, starch solution and the amylase solution. In order to regulate, control the pH, we can use buffer solutions. Buffers. So what we do is, here we are adding amylase to a buffer solution which is placed in a test tube. We have to mix this properly and then after we are adding the starch solution. So we have to start the timer. Then after like 10 seconds, we have to add a drop of solution of iodine in the spotting tile. So we have to repeat the same procedure every 10 seconds until the iodine does not change its color. So after repeating the first 5 steps, we can do this, we can repeat this experiment with different pH, like pH 5, pH 9, pH 14 or pH 7, likewise. So then we can identify the optimum pH value is pH 7. Right. Now that is the end of this part. Now we are moving to the questions. First question, which graph shows the effect of temperature on the activity of enzymes? We know when temperature increases, the enzymatic activity also increasing up to a certain point. Then it should be quickly dropped, right? So what is the correct answer? First one. Second one, the diagram represent enzymatic action. You have to identify which one is the enzyme, which one is the product, and which one is the substrate. We will see what is W here. Which one? And which one is the X? Which one is the Y? We can clearly see, Putha, X is the enzyme, right? What is W? W is the substrate. Then what is Y? Y is the product. You can fill the column, right? Y should be the product. Now there are some more MCQ questions, 3, 4, 5 and 6. So you can try these questions at home. I hope you understood the lesson well. Have a nice day. Thank you.